Yantonakis. Actually, it's Anna Katerina, but she doesn't like it, and everybody should tell her cat.
It's not that I don't like it, it's just most people get it wrong, so I might as well make it simple.
This cat Yantonakis, it's a Greek last name, but she actually, as you can hear by her accent, she has she's British actually. She studied biochemistry and molecular genetics in the Imperial College and University of London, UK. Then she did a postdoctoral stay with Andras Nagy at the Lunenfeld. Is that right? With Andras? Okay, yeah, in Toronto, Canada. And then another postdoctoral stay at the University of Columbia. She has been appointed in the Sloan Kettering for a long time, since the 2004. And she's actually the chair of the developmental biology program after Catherine Anderson passed a few years ago. Is that right?
That is correct.
Okay, so she has more than 160 papers, many of them in some of the most prestigious journals where people can publish, such as Genes and Development, Cell, Developmental Cell, Nature Development, Nature Cell Biology, and has more than 11,000 citations to her work. And I think that Kat is one of the persons that in the field that it's always working in searching for new technology and tools to reveal the secrets of embryo development. And I think she's gonna tell us a wonderful story about how to use the embryo to understand how to the lineage specification, the morphogenesis of tissues occurs during development. Also, how the stem cells originate in the embryo. So, Kat, please welcome. And
again, thank you, Diana, and thank you for the very gracious invitation to talk to you, to present some of my lab's recently published and actually a lot of unpublished work. So what I thought I'd do today, as Diana said, my lab is really interested in how cells regulate their identity and how they sort of coordinate to organize themselves into emergent tissues. We use the mouse model, and this is actually how I know Diana, because we were both instructors on the Cold Spring Harbor mouse course about 10 years ago. And we spent, a month at Cold Spring Harbor together organizing and running the course. And I have to say, it was a real pleasure working with Diana. She was very organized and kept us all organized as well. So what I thought I would do today is really sort of pivoting on some recently published work. I'm going to tell you two stories about where we're taking things. I'm going to tell you about, how we're taking a single cell transcriptomic analysis that we performed recently, to try and understand how many of our organs or internal organs, emerge from the embryonic progenitor called the gut tube. I'm going to show you some of the methods and the technologies that we're now starting to use. And at the same time, I'm really going to leave you with many open questions, because this really is an ongoing project, but I wanted to really share with you how we're taking things forward. At the same time, recognizing the embryonic origin of many of the cell types present in the later fetus, I wanted to share with you another story that has been really pushed by one member of the lab in trying to reprogram cells into different identities. In fact, we're trying to reprogram cells into their sister identity that's present in the embryo. We're doing it in stem cell paradigms and trying to ask questions of stem cell plasticity. And again, we're performing reprogramming experiments and using single cell analytical tools to try and understand the trajectory cells take between distinct identities. And this is, again, another story that is unpublished, but we're hoping to actually submit that within the next few weeks. And it'll be really great to be able to have the opportunity to show some of this new data to you and to perhaps get some feedback. So I just want to take you back to, you know, and sort of a topic of my lab's interest. And let me just try and get a pointer here. Rather than so I hope you can see this laser pointer, which is the blastocyst stage embryo. It's a universal stage across mammalian embryos. It takes about 3 to 4 days to develop from a zygote into a blastocyst in mice and about a week in human. The blastocyst we really like because it really is an ideal self-organizing system. You can take an embryo out of a mother, you can develop it in culture in minimal media, and it will generate reproducibly this type of structure, which is sort of like a hollow spherical structure with 3 distinct cell types, color-coded here in green, in blue, and in red. And it also comprises a minimal number of cells. And for us, this is really great because ultimately, you know, it's a defined time scale, a defined, you know, sort of morphogenesis, and a very sort of minimal number of lineages. We can use genetics, time-lapse imaging, and single-cell transcriptomics to understand the formation of the blastocyst. These are the 3 lineages: the trophoblast that will form the fetal portion of the placenta, the epiblast that will form the majority of the embryo proper, and the primitive endoderm, another extraembryonic tissue like the trophoblast that will predominantly form the yolk sac. And these are shown here with respect to color coding of the embryo proper, which is epiblast derived, and these 2 extraembryonic tissues that are really critical for embryo development, but of course dispensable for adult life. The main focus of, you know, early mammalian development is to specify epiblast, primitive endoderm, and trophoblast, and to segregate them, really to set aside the cells that will form the embryo from those that will form supporting extraembryonic tissues. So my lab for many years now have been very interested in this decision within the early blastocyst stage embryo, where these progenitor inner cell mass cells, shown here in purple, make a decision to either make primitive endoderm or epiblast. We've put a lot of effort over the past 15 years to try and unravel the basic molecular mechanisms that drive this decision, the key transcription factors, the key signaling networks. And I think we've made a lot of progress combining tools in mouse embryology, stem cell biology, and time-lapse imaging. What we know, and it's a lot of work that's been published over the past sort of, you know, more than a decade, and I'm not going to go into details, is that essentially within this progenitor population, there is a group of cells that asynchronously will decide to either become blue or red, and that ultimately you can perform embryological or genetic manipulations that alter, you know, gene expression, or in this case, this is an embryological manipulation that a former postdoc, Mr. Seith, performed in the lab, where he could reduce the size of embryos by half or double the size of embryos. And what he showed was that ultimately you would still generate the right ratio of cells and in the right sort of organization. So essentially what we know is that cells and embryos, even though they can, in this case, one progenitor population has one decision and 2 outcomes, it does so over time. Decisions are asynchronous, and ultimately, collectively, this ensures we now know a consistent lineage composition. We understand details of mechanisms by which cells talk to one another, so that blue cells can then tell the uncommitted population how many of them exist, and ultimately how many red cells should be specified, and vice versa. So really what these types of experiments demonstrated was that within embryos, there is asynchrony in cell fate specification. We actually went on to really drill down on this type of observation by exploiting the recent sort of over the past 5 years advent of single cell transcriptomics. And what we've done, and it's now published, is take these pre-implantation stage embryos, dissociate single cells, load them into a microfluidic chip for encapsulation of individual cells, and these gem beads for barcoding and single cell sequencing and analysis. And this has been really a game changer for this, where we can ultimately isolate cells out of embryos, and also I'll show you stem cell populations, and we can perform single cell transcriptomics. This is the kind of plot that I'm going to show you a lot of. It's called a force-directed layout. Every dot is a single cell. This is a dimensionality reduction type of analysis. And what this shows you is inner cell mass cells, or cells recovered from the inner cell mass. So we've removed computationally the green trifectoderm cells over time, and we can cluster cells with algorithms. This is an algorithm developed by our collaborator Dana Pear's lab called well, we're using multiple algorithms. This is the trajectories plotted in Palantir, and ultimately what we can see here is data from 3.5 to 4.5 embryos, stage embryos, single cells, and they can be clustered to represent distinct identities. Purple is uncommitted inner cell mass cells, and these sit here at the top. And then you can see two distinct arms, epiblast, or cells that acquire an epiblast identity versus cells that acquire a primitive endoderm identity. This is just showing the data in a different way, where we're clustering the data based on the stage of the embryo that the cells were collected from. And you can see the 3.5 day cells cluster together, as do the 4.5 days, by which time cells have made a decision to generate either primitive endoderm here on the bottom right or epiblast on the bottom left. We can also look at differentiation potential, which is a sort of mark of the probability of cells differentiating. And again, it's a computational prediction, and our computational collaborators could predict where cells had a propensity to make a major lineage decision, and that coincides with what we recognize as inner cell mass. And then they can plot this over pseudo-time using this algorithm that they developed. And ultimately, over pseudo-time, you can see that cells are going from this early population to these two divergent outcomes. What I should mention is that this type of analysis really validated a lot of the observations we'd made previously with minimal numbers of markers. And it was part of an analysis, a large-scale analysis, where we put together 120,000 cells, single-cell RNA-seq atlas of the development of the endoderm. The endoderm, I have to say, is my favorite tissue. It's the tissue that will form the extraembryonic endoderm of the yolk sac, and perhaps more interestingly, and I will focus on that on this talk, the endoderm of the gut tube being the progenitor tissue for all our visceral organs, the lung, the liver, the pancreas, and their associated respiratory and digestive tracts. Endoderm is first specified in the blastocyst and then again at gastrulation. And what Sonia Novochin, Beth Kuo, and Vithagard did was isolate endoderm cells from sequentially staged mouse embryos to construct an 120,000 cell transcriptomic atlas that covered all phases of endoderm, all types of endoderm, from pre-implantation to mid-gestation. We collaborated with my colleague Dana Pear in the computational biology program. Dana really is a pioneer of single-cell transcriptomic and other omic analysis. And the first study was really spearheaded by Manu Sethi in her lab. So what our collaborators could do once we had generated libraries and sequenced them from cells of the endoderm of these distinct stages is start to analyze them computationally. What they demonstrated, or they could aggregate all the different libraries, and they could show a trajectory from pre-implantation through to the gut tube post-implantation, and also the extraembryonic tissues of the yolk sac at post-implantation. So essentially what I have drawn here is color-coded over here to the right in the force-directed layout, and you can see how the trajectory splits between the embryo and extraembryonic. Essentially, this type of analysis gave us a number of surprises that I'm going to just overview. The idea being that when inner cell mass make a decision to form primitive endoderm or epiblast, this is meant to be a fixed decision. And then the primitive endoderm, as Diana knows very well from her work, will differentiate into either visceral endoderm that is adjacent to epiblast, or parietal endoderm that receives relatively little attention. The visceral endoderm will generally form the endoderm of the yolk sac. Then this epiblast will mature, and at the time of gastrulation, at around 6 days, it will differentiate to the 3 germ layers of the embryo: the ectoderm, the mesoderm, and the definitive endoderm, the endoderm that is meant to be the germ layer that seeds the gut tube. So this is just a schematic of what a mouse embryo looks like at this stage, and these are the lineage kind of trajectories. What the analysis that we performed of our single cell data showed was that if we plotted 3.5 in red, 4.5 in yellow, and 5.5 data in green, again, single cells, force-directed layout, every dot is a single cell. You could see the ICM of 3.5 bifurcating to these two branches. This is the epiblast branch, as you can see here, colored in red. This is the primitive endoderm branch, shown here in blue. And ultimately what they gave us was a bit of a surprise that we saw a bridge between these two distinct trajectories, which we thought really kind of disagreed with this kind of exclusivity of these two lineages. They plotted this in pseudo-time, and what was surprising was when they plotted computational differentiation potential, propensity to differentiate, there were two regions of increased differentiation potential. The origin of the inner cell mass and also something over here, which we thought was very interesting. It was rooted in the epiblast, and this argued, computationally predicted, that cells of the epiblast over here or here might somehow be differentiating into this endoderm identity. So this is just looking at the primitive endoderm, visceral endoderm propensity versus epiblast. And effectively, this is just highlighting here the second point of elevated differentiation potential, arguing or suggesting that there might be an unexplained or previously unappreciated differentiation of epiblast prior to the onset of gastrulation, and that epiblast might differentiate into endoderm. So essentially, this was the sort of question that ultimately, does epiblast differentiate into endoderm, or does endoderm differentiate into epiblast, shown here in the lineage diagram and here in the diagram of the embryo. The way we could address this question experimentally in the lab was we could use Cre recombinase lines that were epiblast or visceral endoderm specific, and Cre reporters. And this is a reporter that many of you may be familiar with. It's the MTMG reporter that turns green and is membrane bound upon recombination. I'm only showing you the epiblast Cre because this is the one that gave us the interesting result. These are 5.5-day embryos, low mag, high mag. These are 6.5-day embryos. And what you will appreciate is that the epiblast Cre predominantly label cells of the epiblast. But if you look at high magnification, as illustrated here, ultimately you can see a couple of cells labeled within the visceral endoderm. This would actually argue that cells originating in the epiblast actually eventually end up in the visceral endoderm. As I mentioned, when we did the visceral endoderm Cre recombinase with the MTMG reporter, we never saw cells initially in the visceral endoderm end up in the epiblast. So what this argued was that there is precocious differentiation of epiblast into endoderm before gastrulation. And it's schematized here, and this is the direction of the differentiation on the lineage tree and then on the embryo. And I want you to hold that thought, because in this last part of my talk, I'm going to tell you about reprogramming endoderm blue cells to red epiblast cells. And you're going to see something very distinct about the direction of reprogramming, because we're performing reprogramming from blue to red and red to blue. So what this in vivo experiment, prompted by a computational prediction made using single cell RNA-seq data, argues is that there is a rare but still exists a differentiation event from epiblast to endoderm before gastrulation and after specification in the blaster. Okay, yes.
Can I ask you a question?
Absolutely.
The position of those cells is already anterior or posterior, or?
It doesn't. So what you're asking is, are these cells that could be anterior visceral endoderm or
Yes.
not? We don't know, because we never could combine it with a marker, because it was MTMG. We could never combine it with a hex GFP or an ABE marker. At the time we did the experiment, we also didn't have fluorescent in situs working. We now do. So in principle, we could address that question, but from the experiments that we've done, we're not convinced that the cells that are differentiating into a visceral endoderm identity or exiting the epiblast, perhaps because they're not fit to be pluripotent, we don't believe they are specified as a particular subtype of visceral endoderm. Does that answer your question?
Yes, thank you.
Okay. So the other thing that we thought was really interesting was that if we again collated these data across stages and we looked late at mid-gestation, our terminal endpoint, we could actually follow this bifurcation, and there was a crossover very early. You don't see it here because this is taking all 120,000 cells and integrating them into a single plot. But we also saw that at the sort of later sort of gut tube stage, the gut tube was comprised of cells of both definitive endoderm origin, which are embryonic, and this extraembryonic visceral endoderm contribution. This is something we'd seen previously using lineage tracing and live imaging, but we'd only gone until 7.5 days. What these data allowed us to do is to go later and to, in an unbiased computational way, predict the origin of cells across the gut tube. What we thought was very interesting, and I'm going to talk about that a little bit later, but what we thought was also very interesting is we could generate a force-directed layout of the 26,000 single cells, so single cell transcriptomes we'd recovered from mid-gestational gut tube, and Manu Sethi, working with Sonia Novochin, could plot them in a force-directed layout. We could cluster them based on gene expression similarities, and we found these kind of unusual clusters across this force-directed layout. When Sonia looked at the genes enriched in each of these clusters, she recognized that something very striking, that ultimately each of these clusters represented progenitors of each of the organs. And this we thought was very interesting because within this cluster of single cell data, we effectively were seeing an anterior-posterior axis. And when we plotted the clusters across pseudospace, we could actually see the order of these sort of clusters representing organs as early as mid-gestation that reflected the order of organs in the adult. And we think this is really interesting because the mid-gestational gut tube is featureless. And prior to this study, it was not appreciated that cells with mid-gestation already know that they have a bias in terms of forming, thyroid, thymus, lung versus small or large intestine. So we thought this was really telling us that patterning of the gut tube happens very early, and it happens well before organotypic outgrowth. We could identify genes that are expressed along the AP axis of the gut tube computationally. Manu would identify them across pseudospace, and then Sonia, assisted by Beth, would perform mRNA in situ hybridizations to show that the computational prediction could be validated by experimentation. The problem that we face repeatedly by looking at the gut tube is that the gut tube is very deep in an embryo, and it's very fragile. And if you're going to use a marker to look by mRNA in situ at this deep tissue, we had to microdissect the tissue out of the embryo to be able to visualize it. Otherwise, we would get back too much background from the mesenchyme and and all other tissues that are sort of more superficial to the gut tube. So this is what they've done. They microdissected painstakingly entire gut tubes from mid-gestational embryos and then processed them to mRNA in situ hybridization. This is time consuming, but it's the only way to look at endodermal expression until now. So what we've now been doing, and this is really a development that we made in the past year or so, is we've moved to fluorescent in situ hybridizations. They're working rather nicely for us. What this allows us to do is to circumvent having to microdissect tissue. We can visualize in whole mount. We can visualize multiple probes. In this case, we're looking at 3 probes. We can actually go up to 6. And ultimately, we're labeling the entire gut tube by claudin 6, the emergent liver with HEX, and the liver and pancreas with PDX1. Because this is a fluorescent method, we can ultimately quantify the levels of expression. And I'm just going to go back and show this is a three-dimensional reconstruction of a laser scanning confocal image. Every dot is a single mRNA. This is performed in whole mount across an entire 8.75 embryo, around 13 to 15 somite stage. We can use the Claudin to actually segment the entire gut tube because Claudin labels the entire gut tube. And what we can now do is visualize things in 3 dimensions across the structure buried deep in an embryo. What we can also do that we're really excited by is then start to look at multiple probes and combination. So we use Claudin-6, as I mentioned, to delineate the entire gut tube. We can take these types of data and then render them sort of to a generic sort of archetype and then plot gene expression along anterior-posterior axis. We can look at multiple embryos and compare, and then we can ultimately generate a surface of the gut tube. And then from that, we're working with our computational colleagues to generate maps, 3-dimensional maps of the structure that we can then, onto which we can then push on or annotate gene expression domains. So this is where we're taking mRNA in situ hybridization, localization of gene expression representing organs prior to their formation. In these embryos, they're a little later than we RNA-seqed in the first study, and you can see the liver is beginning to emerge from this sort of generic sort of featureless gut tube. So we're starting to go a little later in development, and we're starting to annotate the mRNA in situ localization onto 3-dimensional maps of the structures that we're interested in. One of the things we're also interested in, and we know that the patterning of the gut tube arises through cell-cell interactions, both across the gut tube between cells of endoderm, but also between endoderm and mesoderm. We, in our original study, had reported the localization of key genes or targets of key signaling pathways. What we now can do is we have RNA-seq data for mesoderm that is associated with the endoderm, and we can plot the clustering and RNA-seq data for both the endoderm and adjacent mesoderm, shown here in this force-directed layout, where we can go from thyroid to large intestine, from pharyngeal to sort of pre-Semitic mesoderm, and then we can make predictions about ligand receptor interactions across the AP axis between mesoderm and endoderm. So we think this is really where we're starting to push things, to look how cells are cross-talking and making computational predictions regarding signaling. And ultimately, we're trying to build all of this information into a 3-dimensional sort of atlas of gene expression and sort of cell identity. So I just wanted to also go back to this idea that this is the mid-gestational sort of embryo. This is a 3-dimensional reconstruction of real data where we've taken serial sections, every dot of this spinning gut tube. So this is the gut tube computationally isolated from sequential transverse sections taken through one of these staged embryos. Every dot is a nucleus, and effectively what we're doing, if I can just make it play again, is it rotates in blue, then it rotates in green and gray, so that in green are these extra embryonic cells, and then finally it rotates color-coded so that you can see the different organs color-coded at mid-gestation at single nuclear resolution. So one of the things we found was very surprising was that the mid-gestational gut tube comprises cells of 2 distinct origins, predominantly definitive endoderm, but also, at least enriched posteriorly, these extraembryonic cells that many people, didn't believe would actually contribute to the gut tube because they're meant to be an extraembryonic lineage. This was shown computationally. And what we thought was very interesting was that when we looked at these cells, irrespective of their origin, they would express the same markers. So markers such as IRX, PPY, PDX, that are, for example, PDX is a famous marker associated with the pancreas. It's expressed both in cells of definitive or epiblast origin and also gut endoderm cells of an extraembryonic or visceral endoderm origin. So we thought that ultimately cells of two distinct developmental origins come together into one embryonic structure and have this transcriptional equivalence. We looked specifically for genes that would distinguish cells of definitive endoderm, so embryonic endoderm versus extraembryonic endoderm. And we couldn't find any genes that were statistically significantly enriched in cells associated with definitive or embryonic endoderm. However, we did find a few genes, about 4 or 5, that were enriched in the extraembryonic or cells of extraembryonic endoderm. So these were genes that are associated with the extraembryonic endoderm and would argue that cells of an extraembryonic endoderm origin present in the mid-gestational gut tube have a memory of their past history, that they were extraembryonic, not embryonic. One of these genes is a sort of transcription factor in an atypical homeobox gene called ROX5. This is the computational prediction of our sort of 26,000 single cell transcriptomes from the mid-gestational gut tube, color-coded by embryonic pink versus extraembryonic origin. This is the in silico expression of ROX5 predicted by the computer. This is a ROX5 whole mount mRNA in situ hybridization, where the gut tube has been microdissected out of an embryo, and you can see this enrichment posteriorly of ROX5 and this kind of punctate distribution more anteriorly that kind of agrees experimentally with this computational prediction. And this is a reporter knock-in that we've made in the lab, whereby we've used gene targeting in embryonic stem cells to introduce an H2B GFP to the endogenous ROX5 locus. We've then isolated embryos, we've imaged them in whole mount. This is a 3D reconstruction of a confocal Z stack. It's been stained with FOXA2, a marker of the endoderm that delineates the entire gut tube. And you can see ROX5 that is nuclear GFP localized, associated with the yolk sac, a known site of ROX5 expression and a visceral endoderm contribution, and then this distribution that throughout the gut tube enriched posteriorly. So this knock-in reporter faithfully recapitulates the mRNA in situ hybridization data, which again validates the computational prediction. So armed with a tool such as this, we could then go in and further modify the ROX5 locus, and instead of knocking in an H2B GFP reporter, we could put in an inducible Cre. So the experiment we can then, and we are now doing, and I'm going to show you the first data because I think it's really interesting, is we can ask the question of what is the eventual fate of these extraembryonic cells within the later gut tube. The question being, well, we see them at mid-gestation, and many people will say, well, you see them at mid-gestation doesn't mean they persist. The big picture question is, do they persist? Do they incorporate into the organs as they form? And do they then persist to the adult animal? Do they persist during normal homeostatic conditions? And what happens to them during disease conditions? So do they play a functional role? And under a stressed or disease or pathogenic condition, do they behave the same way as their neighbors of an embryonic origin? I'm not going to answer all these questions today because those experiments are ongoing, but this is the experiment that we wanted to do. Can we ultimately fate map this extraembryonic visceral endoderm? I've shown you that cells persist in the gut tube. What happens later? The way we do this is we use an inducible Cre, and this is a ROX5 Cre RT2 mouse that we've generated. So what we did was we identified ROX5 from the single cell RNA-seq data. We validated it by mRNA in situ hybridization. We made an H2B GFP to demonstrate the localization of the reporter, and then we could make a similar or use a similar targeting strategy to generate a Cre RT2. We could then pulse early and chase late. The reason we do it by a pulse chase, not a regular Cre, is that many markers expressed early in the endoderm are reactivated later in the gut tube. And we wanted to circumvent any issue with reactivation, that a reviewer could say to us, oh yes, well, you only see cells in the emergent pancreas because they're reactivating a gene expression of a gene that was expressed early. So we would pulse early, chase late. recognizing that these late stage embryos are really difficult. They're a pain to section. You could sit there for weeks sectioning everything transversely. What we have chosen to do, and this was really spearheaded by James Muller, who's associated with a microscopy sort of cluster that we've established in our department, is we could use tissue clearing methods. So we can take embryos, we can fix them, we can optimize them for various tissue clearing procedures that are compatible with either immunofluorescence or GFP labeling. We're using a variation of iDisco, and it's iDisco Plus. And ultimately, what this allows us to do is to take an embryo at an advanced stage, to label with immunofluorescence or with a GFP reporter distinct features, and we can visualize them in 3 dimensions. So this is an embryo, 12.5 days. We've labeled it IF with antibodies against SOX2 and NKX2.1. You can see the SOX2 delineating the brain and spinal cord. You can also see it delineating from the mouth through the respiratory tract, and you can see the esophagus all the way to the stomach, and then you can see the thyroid, the thymus, and the lungs in beautiful single cell resolution. If I were to zoom in, you'd see this to be nuclear localized. These are tissues buried deep within embryos. So this type of tissue processing and clearing technique allows us visual access to structures that are really very difficult to get access to normally. You'd have to section, and then you have to get the right section, and you can't compare multiple embryos because you can't always orient them the same way in the blocks for sectioning. So this type of clearing is used by Beth and Sonia, coupled with their ROX5 Cre-RT2 mouse. They're labeling the gut tube with an EpCAM antibody shown here in teal. They're cellular descendants of the extraembryonic or visceral endoderm labeled here in pink, and this is a TD tomato. It's a TD tomato Cre reporter. What you can see here in this embryo is the intestine. The pancreas is beginning to form the dorsal and visceral and ventral branches. The liver is starting to bud, and these are the lung buds. You'll see they're on either side. This is a whole mount preparation. It's actually a light sheet microscopic image that's been 3-dimensionally reconstructed, and it's going to spin. It's going to spin twice. The first time you'll just see the gut tube and the details of the respiratory and digestive tracts as they're starting to emerge. As it spins, you'll start to see some pink cells that will be localized, I hope to convince you, specifically within these endodermal organs. So this is spinning. You can see this is truly 3-dimensional. We're looking deep within the tissue. And then as this spins, around now, you're going to see the emergence of these descendants of this extraembryonic tissue. Predominantly posteriorly in the intestine, but you'll also see them, a few within the pancreas, a few within one side of the pharyngeal endoderm, not the other. So this is new data, but it's arguing that these descendants of an extraembryonic origin persist, and they are exclusively localized within the gut endoderm, predominantly posteriorly. Okay, this is an ongoing study, and I'm actually going to shift gears now, if you don't mind, for the final part of my talk. And I know I'm running a little bit late, so I'm going to talk fast if I can. So all this work that I've shown you so far, or predominantly shown you so far, is unpublished. It's ongoing, but I thought I would share it with you because we think it's really exciting. It's exciting because we're embracing new technology to try and understand, cell identities in 3 dimensions. And we're using new methods to try and probe lineage relationships and the fate of cells, for example, extraembryonic endoderm, that were believed to be exclusively extraembryonic. So as many of you know, the blastocyst is a universal stage of mammalian embryogenesis. There are 3 cell types, as I alluded to begin with. And again, I'm sure many of you know that the blastocyst is a unique stage from which you can derive cells that represent each of the 3 lineages. ES cells, very famous, represent the epiblast. Zen cells, extraembryonic endoderm cells, represent the primitive endoderm in blue, and TS cells represent the trophoblast. My lab are really, to be very frank, very interested in ES cells and Zen cells. We don't do much on TS cells. So one of the questions we wanted to ask, and this was really a heroic effort taken on by Vidar Garg, a student in the lab who graduated in January and has remained as a postdoc until we submit his paper within the next month. He wanted to ask, can we convert ES cells into Zen cells and Zen cells into ES cells? And can we, by converting, learn something about the trajectory cells are taking as they interconvert between distinct fates? These are identities that are represented in the embryo and can also be captured in culture. So if we force conversion or reprogram cells from one identity to the other in culture, what trajectories do they take? And do the trajectories that the cells in culture take represent anything that we would see in an embryo? So actually Vidar knew, and we've done some work in the lab previously, that a single transcription factor, when overexpressed in ES cells, is sufficient to drive them to a Zen-like state. Interestingly, there is no report as yet of reprogramming Zen cells to IPS cells using OKSM or any of the other Yamanaka-like factors. So Vidar wanted to try this. He exploited two comparable systems where essentially he had DOX-inducible GATA4 versus a DOX-inducible OKS label with a cherry. We collaborated with this for the study with our colleague Effie Apostolou, who works across the street at Cornell University. She's actually a reprogramming guru. She came out of the lab, she did her postdoc in the lab of Konrad Hochedlinger, and she started her own lab over at Cornell about 6 years ago. This is when Vidar came to the lab as a student. We thought this would be a really cool experiment to do, to look in vitro at interchange, the plasticity of cells representing states present within the embryo. So effectively, when Zen cells can be reprogrammed, Vidar showed very early on, into ES cells. So you can see M-cherry is the transgene construct, and then also in his Zen cells, he had an OCT4 where he had knocked in a GFP. So this is the endogenous lockers where we can see GFP associated with OCT4 that labels pluripotent cells. So what Vidar demonstrated was he could take Zen cells into an IPS state. But what was surprising was it took 3 to 4 weeks and was less than 1% efficient. It was highly inefficient. It's actually one of the least efficient IPS reprogramming experiments. I don't want to go into details, but essentially he was making bona fide IPS cells by virtue of markers. His cells were losing in blue Zen or primitive endoderm associated markers, and they were acquiring pluripotency associated markers. He could make chimeras from his IPS cells, and he could demonstrate that by contrast to Zen cells, his IPS cells would contribute exclusively into the epiblast of the embryo, demonstrating stringently that he had generated IPS cells. He performed a number of assays that I don't have time to tell you about, where he could show the trajectory of different marker genes and showed that there was a bottleneck whereby the cells, or it was the inefficient step, was the loss of the Zen program and activation of the pluripotency associated program. He also performed experiments where he could take ES cells into Zen or iZen cells using this overexpression of GATA4. Same thing, opposite direction. When he did this experiment on the left, he found something really very interesting. And this is a time-lapse sequence where he actually has a cherry marker for his transcription factor that is overexpressing, and then a venous marker that is endogenous to a primitive endoderm gene GATA6, and he can show that there is successful reprogramming and expression of an endogenous marker by virtue of venous. He could show this ultimately by flow, but I think what is the most important point is that if he takes the reprogramming in this direction, it takes 3 to 4 days and is 99% efficient. Again, we've done lots of experiments where you can look at the trajectory, you can look at the bottlenecks, and this is what we think is really kind of interesting. You go from Zen to ES, very slow, highly inefficient. ES to Zen, highly efficient and very quick. Remember what I told you about the embryo. When we looked at the epiblast versus primitive endoderm or visceral endoderm trajectories, we saw rare but occasional events where epiblast cells would go into the visceral endoderm. We never saw the opposite. And we think that this may be reflected here in culture, where we have this very efficient differentiation, versus a highly inefficient and very slow one. So what we did, having sort of generated these data, is perform a single-cell RNA-seq experiment for both directions of the reprogramming. We went from start to end states, and then we went from 3 states in the middle. And ultimately, we could compare this with our single-cell RNA-seq of the embryo, which is the first time we can look at trajectories directly between embryos and ES cells. These are the force-directed layouts of the ES to Zen. What's really interesting is this is a very simple structure. It's a linear trajectory, and it reflects a highly efficient process. And here we're just mapping the cells from the different time points that comprise this entire sort of population. What we did was we recovered single-cell transcriptomes from ultimately 10,000 cells from each state. So a total of 100,000 cell transcriptomes, 50,000 for each. So 50,000 for ES to Zen and 50,000 to Zen to ES. By contrast, this is the very inefficient and very slow procedure, Zen to IPS. It's a very sort of protracted trajectory. And I'm going to tell you a little bit about some of the details here. There seem to be some bottlenecks. Here, there are no bottlenecks because it seems to be 99% efficient and very quick. You can see the timeline is also very distinct here. So ultimately, this is differentiation potential. It's actually 0 because there is no other alternate fate that a cell can take in the ES to Zen trajectory. There's a start and the end state, and over 99% of the cells will do that. If we go from Zen to IPS, there's something very interesting. In the interest of time, what I want to draw your attention to, that we found 2 terminal states along the trajectory prior to reaching IPS, meaning that these are actually sinks where cells can settle in, and it takes some sort of activation energy to get them out, to get cells all the way to IPS. So you can think of these states a bit like this. We're going from Zen to IPS, and there are states in between that seem to sort of be attractor states where cells are settling and not efficiently moving forward. If you were to draw the same from ES to Zen, it would be a straight line going down with a very sort of quick trajectory. So we can actually look for differentiation potential, and we can look for markers. This was interesting because we can look for primitive endoderm and epiblast markers in this Zen to IPS trajectory. And what we noticed was only after cells could exit T2 are they truly losing the primitive endoderm associated gene expression, which is again what our limited marker analysis was telling us. What this single cell analysis also informed us is that ultimately OP4 is the first marker that is associated with pluripotency that is activated in cells that are already in this T2 state. And as they leave the state and go up this way to reach the terminal T3 or Zen state, they will activate NANOG and SOX2. So there really seems to be an order in the activation of pluripotency associated markers. What we think is interesting, and we're still doing some work trying to figure this out, is we've merged the trajectories from Zen to ES and ES to Zen. And it appears as though they actually reveal that similar states are traversed. So from going from blue to red and red to blue, you seem to be going through the same intermediate state, which we think is very interesting. And it's something we're looking at the moment. And this would argue that the differential conversion rates and efficiencies probably reflect the relative plasticity and perhaps something to do with the epigenome of cells rather than their transcriptomes, if the similar transcriptomic states are being traversed in both directions. Interestingly and importantly, we wanted to map everything back onto the embryo. So we thought this was really kind of important. So we could take our trajectories, we could bin them from early through to late, from blue all the way to yellow, and we could take those states and project them back on to the embryo single-cell RNA-seq data. This shows the data from 3.5 to 4.5 days that I've shown you earlier, where we have ICM cells that bifurcate, where cells make a decision to form epiblast versus primitive endoderm. And sure enough, the Zen state is rooted in the primitive endoderm. And as cells start to convert, they go up the primitive endoderm branch, and then interestingly, they jump all the way through to the epiblast and then go back down. We were surprised that we never saw cells enter the ICM state. And this again is something we can discuss. And I think there's many people have tried very hard to try and capture in vitro an ICM-like state and failed. You can either get Zen cells or you can get ES cells, but you can't get a bipotential progenitor in culture. And we thought this was very interesting that they would jump this way. We also did the same type of mapping where we took ES to Zen. Again, this is the very efficient, very short state, but again, and rather gratifyingly, ES cells map to the end point of our sort of Zen to IPS trajectory here, and we can follow it all the way through. Again, they jump, they don't bypass an ICM state, but they jump from one branch to the other branch. So we're getting the jumping in both directions, and in each direction, cells are bypassing this inner cell mass uncommitted progenitor state. So this, we believe, is one of the first times where we have plotted in vitro trajectories back onto in vivo trajectories. And again, they're telling us something very interesting, that the same states are being traversed, whether we're reprogramming from epiblast or primitive endoderm to epiblast, and both of these trajectories are avoiding the inner cell mass.
Excuse me,
So
may I ask
this is
you
my
something?
final slide. I'm sorry.
Yes, may I ask you something?
Absolutely.
The last slide. Yes, here you are showing that you never get inner cell mass identity when you go from Xen to IPS.
Correct.
But you previously mentioned that if you make an aggregation with morulus, then all these IPS goes into the inner cell mass.
No, they go into the epiblast.
They go to epiblast, okay.
So they will incorporate into the inner cell mass, but they have an epiblast differentiation potential, so they are exclusively restricted to the epiblast. And I didn't show you because we've actually done the experiment with Zen cells, unreprogrammed and reprogrammed, and our Zen cells will go exclusively to primitive endoderm derivatives, visceral and parietal endoderm, and then our IPS or our reprogrammed Zen cells will only go into the epiblast.
So you would expect that epiblast stem cells would behave exactly as these reprogrammed parent endoderm, right? The Xen going to IPS will show a prime state different from the...
So the Zen going to IPS does not show a prime state. The prime state would actually probably be down here. And we've actually, I'm not showing it here, but we have mapped epiblast stem cells to our in vitro RNA-seq data, and they actually fall at, they fall at 5.5 day epiblast, not 4.5 day. Okay. Whereas IPS and ES cells are rooted around the trajectory at 4.5 days.
Great, thank you.
You're welcome. Okay, so this is my final slide where I just wanted to bring everything together. I've shown you this, which was RNA-seq data, single cells isolated from inner cell mass of blastocyst stage embryos. You could see that cells are sort of asynchronously over pseudo time making this binary fate decision that we could then visualize and validate through experimentation. These experiments, when we layered on the 5.5 day data, they showed us this unusual kind of rare differentiation event where ES cell or epiblast would differentiate into endoderm. It would exit pluripotency. We validated this computational prediction experimentally, and it holds true. Because I have to say, when our computational collaborators first told us this, we said this is not believable because this goes against the textbook of developmental biology. And they said, have a look. And we said, no, you've done your analysis wrong. And then we said, okay, let's do the experiment to prove them wrong. We did the experiment, and in fact, we were wrong, because our understanding of developmental biology and lineage restrictions was not as robust as we thought it was. And we managed to perform experiments that would validate a computational prediction that we actually didn't believe. So what I also showed you was in our reprogramming of ES cells to Zen cells, and this is the first time I believe that anyone has looked at bidirectional reprogramming between two bookended states. And what these in vitro data demonstrate is that one direction is highly efficient and very rapid, and the other is highly inefficient and very slow. And we think that the ES to Zen somehow represents an in vitro counterpart of this differentiation event. That ultimately ES cells will sort of, continue to a 5.5 day embryo, and at some point when they lose or they're not fit to be present in the epiblast, or they're not fully pluripotent, they will differentiate, and the sort of the default must be an endoderm identity, and this will get them out of the epiblast and into the endodermal layer. And we've got data where if we knock down the endodermal program in Zen cells, trying to look mechanistically at what is stalling this, we actually notice that we can improve the efficiency of reprogramming for Zen to IPS when we go in and CRISPR abrogate the Zen program. So we could schematize what we're sort of looking at a way a little like this, that ICM cells will make epiblasts and they'll make primitive endoderm. But as you see here, and this is reflected in a pseudo-temporal plot where the primitive endoderm is further away from the inner cell mass than is the epiblast, perhaps the primitive endoderm is further down the hill than the epiblast, so that epiblasts can go to primitive endoderm quite easily, but primitive endoderm can't go to epiblast as efficiently. And I always actually like to say that these cells are going up the hill backwards, because they really are, rather than sort of differentiating, they're kind of needing to go up backwards. And this is where at the moment we have generated and we're currently analyzing single cell attack-seq data to see whether there is some aspect of the sort of chromatin accessibility at key loci that is somehow preventing this state readily reverting to this state. But by contrast, this state will easily sort of differentiate into this state. I think this is my last slide. I think I mentioned most of the people involved in the generation of the data I spoke about today: Vida, Beth, and Sonia. I wanted to give a shout out to our collaborators. Effie Apostolou, as I mentioned, has been really critical for the reprogramming. She is a reprogramming sort of guru, and we've worked quite intensively with her lab over the past couple of years. Mark Lewandowski, who is someone that Diana Escalante knows very well, has been a collaborator along with Matt Anderson in his lab. So they're at the NCI Frederick, where Diana did her postdoc. And Mark is the person who really brought to our attention and trained us for the fluorescent in situ hybridization technique that I showed you. I was at a meeting where Matt Anderson, Mark's senior scientist, presented data. I almost fell off my chair. It was incredible. And I contacted Mark and I said, well, we need to learn this technique. And someone from the lab visited Mark's lab, and we have been collaborating ever since. In terms of the single cell transcriptomic analysis, and now also the single cell attack analysis, we've been collaborating with our colleague Dana Pear here at Sloan Kettering. Manu Sethi did a lot of the initial RNA-seq analysis, and Esther Wershoff is really taking over, performing the RNA-seq, some of the attack, but also some of the image data analysis, where we're taking these three-dimensional fluorescent mRNA in situ images from sequentially or similarly staged embryos, and Esther is generating a generic gut tube atlas, which we're decorating with gene expression profiles. I think I'm going to stop there. My time is up, and thank you for listening.
We're open for more questions. I have a couple, but I will have to.
And I apologize for running late. I just looked at my watch.
No, it's okay. It's okay. We're still on time. I don't know if there are some questions, extra questions. Ivan Velasco.
Yes, thank you. I really enjoyed your presentation. I don't know if I missed it, but I noticed that you didn't use MYC in your reprogramming cocktail.
We didn't.
And what was the reason for that?
The reason is very straightforward. The reprogramming cocktail was actually a cell line that we derived from a mouse that Effie Apostolou and Matthias Stadfeld had generated in Konrad Hochedlinger's lab. This is the mouse that they use all the time. It's an OKS with a cherry rather than a MYC, and it seems to reprogram quite efficiently for fibroblasts. I have to tell you, we've also tried the Zen to ES reprogramming with viruses and with MYC, and it doesn't change the efficiency.
Okay. The other question is, you put in your last scheme ES and epi cells.
Yes.
What happens if you express GATA4 in naive ES cells, prime ES cells, and epiblast stem cells? Do they all differentiate that efficiently?
They all do, and that's actually a really interesting question that we have not looked at systematically, but we do want to look at. So there's a lot of hand waving and a lot of studies that have not been performed rigorously across labs. So for example, Janet Rossent has a study that she published in 2017, 2018 in Cell Stem Cell. Cheryl Seguin is the first author. They misexpressed the GATA factor in human ES cells, and they showed that those human ES cells will generate definitive endoderm. By contrast, the idea is that if you express in naive mouse ES cells a GATA factor, you generate primitive endoderm or a Zen-like state, which is a little distinct from a definitive endoderm-like state. And the way I interpret these data is, depending on the state of pluripotency a starting population is in, will dictate the ultimate endodermal identity that is achieved through the misexpression of a GATA factor. Now that we have single cell omics type approaches working, I think the experiment we need to do is to take mouse and or human, naive, formative, intermediate and prime cells, do the misexpression and ask what is the outcome. Are you changing the type of endoderm you're forming depending not on your transcription factor that you're misexpressing, but on the starting state of the cells? So I think that's a really good question that people can hand wave about, but no one's done the definitive experiment. And I think we now can, because now we can be unbiased, we can generate single cell transcriptomes, and we can map them precisely back onto embryos.
Thank you very much.
Thank you.
We have more questions. I don't know if we have questions in YouTube, Violeta?
No, so far there is no questions in YouTube.
I have a couple of questions. It seems to me a little bit counterintuitive that the expression of this ROX5 transcription factor, it's in the posterior part. It's more posterior than anterior, just because in the posterior part, it's where all this movement during the gastrulation it's happening. So it's like how do you envision the movement of the cells or not movement of the cells during this process, which is very dynamic?
This is very dynamic, you're right. But I think what I think this reflects, and I'm just going to stop here. So this is real data. This is an experiment where, let me see if I can, where we sliced embryos. We did a fate mapping experiment, and we could trace at single cell resolution cells of visceral of extraembryonic endoderm identity. What this is showing you is that they're enriched posteriorly. And this is in agreement with the single cell data and with the ROX5 reporter. So this is an experiment where we took a TTR creed that you're familiar with. We can't use it beyond this stage because TTR becomes activated across the entire gut tube after 9 days, it's around 19-20 somites. But what it does early, it works as a very nice reporter for cell fate of extraembryonic endoderm. This gives us an enrichment posteriorly. So this is why I think that this ROX5, I'm not surprised it's posteriorly enriched. I think this is far, these events are later than the kind of gastrulation movements at the posterior.
Oh, I see.
This is really when the gut tube is already closed posteriorly. So I would say, well, yes, if we're talking about 7.5 days, it is unusual. But I think by the time the gut tube closes, and by the time we're at the 13 to 15 somite stage, which is essentially our E8.75, then I don't think there is movement. This gut tube is a nice sort of cuboidal epithelium.
Okay. And the other, it's kind of, well, you already answered about the difference in the epigenetic marks in the different states between the SEN and the epiblast cells. But the other, how do you think you can capture the inner cell mass state now knowing all the transcriptomics in this?
So I guess my question is, you know, there are these expanded potential stem cells, whether you believe them or not, or you know, cells that seem to be tripotent or multipotent. The question is, should we expect to be able to capture the inner cell mass state? So could the inner cell mass be, as many people have modeled it, including ourselves in collaboration with Jordi Garcia-Yalvo over at the University of Pompeu Fabra in Barcelona. He's done some modeling for us, and you know, people will either, let me just go back to this, where am I? So effectively, you know, people can go back to this, and some people will call this a trisstable state, where there are 3 states. Other people will refer to it as bistable. You're either primitive endoderm and epiblast, and this is an unstable state. So what you're saying is, can we capture this state in culture, whereas this is represented in Zen, this is represented in ES? We've discussed this a lot in the lab. I don't know whether we should expect to be able to capture this state readily, and not have any cell that we capture that has this identity to want to make a decision. I don't know, but again, I think it's going to be difficult perhaps to envisage a manipulation that will allow us to capture this state, but I could be wrong.
I hope you are wrong. That we sometimes can capture exactly that state.
Right.
Yes.
Because the idea being, then you would expect a stem cell, the self-renews indefinitely, when put back into the embryo, will only contribute to epiblast or primitive endoderm derivatives, and will do so kind of equally.
Mm-hmm. Exactly.
And not to trophectoderm.
We have a comment here in the chat, a very exciting talk. Thank you so much, from Itzel Alejandra.
Well, thank you for listening, and thank you for staying on past the hour.
No, thank you very much. It was very exciting talk indeed.
Thank you, Kat. Well, if we don't have extra questions.
I have another question.
Eric, yes.
Thank you. So the first one is about in your single set data, you can study the subpopulation within your populations. You already did that?
Yes.
And is there a common trajectory that shows another pathway between one subpopulation?
You're talking about the in vitro reprogramming experiments?
No, I'm talking about single state data. From your clusters, you had 3 subpopulations: the epiblast, the primitive endoderm, and the inner cell.
So what we've done here, so you're talking about this kind of data?
Yeah.
So we've removed the trophectoderm, which would actually branch off on the side of the epiblast. But these are the only trajectories that one can see. Actually, at 6.5 days, the epiblast will generate mesoderm and ectoderm, but we didn't capture those cells. But ultimately, up to 5.5 days, if we exclude the trophectoderm, these are the only trajectories.
Okay. And
So
I have a second doubt. This might because I maybe missed it from the presentation. So at the beginning of your single cell analysis, you use a gene marker to characterize your populations between epiblast, inner cell mass, primitive endoderm, or?
That's actually a very good point. What we did was we microdissected the endoderm from the epiblast to begin with. Well, after 5.5 days. So 5.5 days, we took everything. We don't need gene markers because for the isolation, we can use gene markers to computationally isolate one cell type from the other from complex populations. So at 5.5 days, we took everything, except for the parietal endoderm. So we took all of, this. Because we can't microdissect easily. At 8.75 by contrast, we microdissected the gut tube. And then what we could do was, because we know there are certain genes that are endoderm specific, we can use those to isolate the transcriptomes for endoderm cells and discard the mesoderm. This is how we actually put together the mesoderm and endoderm crosstalk analysis that I showed you later on, because ultimately we knew who was mesoderm and who was endoderm. So what we actually, what I didn't tell you is initially when we dissected, the gut tubes, they had a layer of mesoderm around them. We could computationally remove mesoderm because we knew, for example, they don't express FOXA2 or Claudin-6 to enrich in the endoderm, and this is what we ultimately focused in on. But then we could reintroduce the mesoderm because we knew it was the population associated with the gut tube that did not express gut tube markers. And then we could also map it pseudospatially because we had a pseudospatial axis on the for the gut tube, and we knew mesoderm that was sitting adjacent to it. We could actually map mesoderm again across the pseudospatial axis. What is also interesting, and I didn't allude to, so anyone that looks at this will see that anteriorly there seem to be some cells kind of crossing over between mesoderm up at the top and endoderm. And this is something that really is intriguing us, whether there are cells that are jumping from the mesoderm into the endoderm or vice versa. Because again, these populations, they're quite separate, but here there seems to be rather a lot of clustering in between. Okay, and I don't have an answer to that question. It's something that we've noticed that we feel at some point we should follow up on.
Thank you very much.
Well, you have changed several times what developmental biology books said, so I will not be surprised that that's also the case, that like these cells like jumping from the epiblast to the visceral endoderm, it will be really mind-blowing if they have some instructed
I don't think they, to be honest, I don't think they do have an instructive role. I think what it is, it's almost like the endoderm is the garbage bin, dare I say, even though I like the endoderm. And I think it's the cells that aren't fit, it's almost like a cell competition thing, that the cells that aren't fit to really be epiblast or pluripotent perhaps are removed from the population. The best way to remove cells from the epiblast, you either throw them into the cavity or you just remove them into the endoderm. And all they have to do is, so that's why I feel that, do they just precociously differentiate into the endoderm prior to the onset of gastrulation, and then they just, are taken up by the adjacent epithelium and removed, because the most important population, dare I say, is the epiblast. And the epiblast has to be poised to differentiate in a very highly regulated way. If a cell is unfit to do so, you presumably have to remove it. You can either kill it, or could you just, if it has a propensity to precociously differentiate, you just, sort it out into the neighboring layer.
Yeah, that's true. But what if not? You will have to do the experiment.
It has to do, there's lots of experiments to do.
Yeah, all right. Well, if we now don't have more questions, I'm very thankful, Kat, that you were able to join us for this institutional seminar. It was incredible to see how everything is moving on in the field, and thank you very much.
Thank you. Thank you, everyone. Thank you for the invitation, and thank you, everyone, for joining and staying on. And apologies for running late.
Thank you.
Thank you.
Fantastic
Bye.
talk. Thank you. Okay, there are some comments in the chat. Okay. All right.
Sí. I know.
Susanita, Gusanita.
Yes.
There
Oh,
she
thank
is.
you very much. Yeah, it was fantastic. Thank you. Thank you very much.
Thank you. It was really great to visit, albeit virtually.
No. Next time you will be here, I'm
In
sure.
person, yeah, please.
Hopefully, maybe
Pero bueno,
next
así
year.
está bien
Yeah.
también.
Hopefully next year, yeah. So are you allowed to travel everywhere in the
No, we're
USA?
not. So we
No.
can't do any business travel.
Oh.
At this point, we can only do personal travel.
Oh, cool. Yeah.
And what
So-
about yourselves?
Say it about? Say
What
it
about
again?
yourselves?
No, we don't have restrictions here.
Okay.
It has
We have
been
many restrictions.
no here. Well, you didn't have any with Trump because everything started to get better with Biden, no?
I think they're getting better, but there's some way to go.
Yeah, I know, but well, here I cannot say the same. We are in the Trump state. All right, so see you and thank you very much. And
Thank you all.
let's Zoom later with
Okay,
with Sonia
absolutely.
and your group. It will be great. And I
Okay.
saw that you are also collaborating with Rocio.
Well, Rocio has joined my group after the passing of Catherine, and I don't know whether you guys are aware, but she's on the job market. So I've been encouraging her to cast her net wide. I think she'd be great if you guys have any positions.
I know, yeah. Okay, so ask her to give me a call or I'll
But
write to her.
I've told her to. We just now put together her package.
Oh.
So you should contact her. I'll tell her again to contact you, but I told her that she should, contact everyone to make sure she is, people know that she's on the job market.
Yeah. Yeah, I will contact her. And the other, it's, well, I will ask her because Catherine had my mice. So, but I don't know what was the fate of the colony. So, okay.
No, definitely ask her, because this is very good timing, because I think it was yesterday that she showed me all of her research statement, CV, etc. And she's putting in applications right now.
Okay, perfect.
Okay,
Thank you
sounds
very much.
fabulous. Thank you all.
Thank you, Kat.
Okay.